everybody. Welcome once again to our Expert ALS talk series. And want to just mention one thing. You're our teammate and we're your teammate. So let's get that really clear up front. Thank you. Without you, we wouldn't be here. We got a special prize. This is a prize and it's coming to you tonight. We will not only have one presentation, but we have a second one. And the second one is very short, but it's an intro into our group, our scientists, our people in Argentina who are working on generative voice AI. So don't go away. Magic is in the air. It's not magic, it's hard work. So welcome. Turn it over to you, Casey.
Hi, everyone. Nice to see you all tonight. Our speaker for tonight is going to be Dr. Nader Yaghoubi. He is the co-founder and CEO of PathMaker Neurosystems. He has over 20 years of experience in the creation, operations, and financing of life science companies, and he established PathMaker to pioneer a new approach to restoring damaged neural pathways. At PathMaker, he serves as a PI on NIH-funded stroke trial and a recently launched ALS trial funded by MDA, and he has led the company to a number of international awards and prizes. He previously co-founded and served as CEO of Symbiotix Biotherapies and was the first employee at Selecta Biosciences and BIND Therapeutics. Earlier, he was a zero-stage capital and completed an internship at Advent International. He completed a fellowship in Health Care Entrepreneurship at Boston University and served as a resident in the Department of Surgery at Lenox Hill Hospital in New York. Dr. Yaghoubi received an MD and Ph.D. in Molecular Pharmacology from the combined degree program at Boston University School of Medicine. Please join me in welcoming our incredible speaker for tonight, Dr. Nader Yaghoubi.
Thank you, Casey. I really appreciate that very kind introduction. It's wonderful to be here tonight, and I'm honored to have the opportunity to introduce PathMaker and our technology to everything ALS and the membership. And I'm pleased to be able to discuss the recent clinical trial that we have launched for ALS. I'm Dr. Nader Yaghoubi, and I'm co-founder and CEO of the company. Our work has been focused on developing a platform for non-invasive treatment of ALS using motor neuron hyperexcitability suppression. At PathMaker, we're pioneering the restoration of neural pathways to treat neurological disorders. We're a near commercial stage neuromodulation company. We're developing non-invasive devices for treating serious neurological disorders. And our initial focus is on ALS and stroke. So what is neuromodulation? I thought I'd start there given the technology focus of your group. Neuromodulation refers to the use of electrical or electromagnetic stimulation of the human nervous system to modulate physiological processes. There are invasive and non-invasive forms of neuromodulation, and they're used to stimulate either the brain, spinal cord, nerves, or end organs for therapeutic purposes. Now, neuromodulation is distinct from brain-computer interfaces, which I think your group is familiar with, which are in development to drive external devices or move muscles. However, both are considered forms of neurotechnology. A few examples of neuromodulation are deep brain stimulation, which is used to treat symptoms of Parkinson's disease. There's sacral nerve stimulation, which is used to treat incontinence, and there's transcranial magnetic stimulation, which is a non-invasive platform to treat depression and OCD. We've been pioneering multi-site direct current stimulation, which is a non-invasive approach to treating motor neuron hyperexcitability disorders. The first such disorder that we began to focus on was muscle spasticity, which is a state of painful muscle contraction. Spasticity is a debilitating condition associated with motor neuron hyperexcitability. It's found in numerous neuromuscular diseases and neurological disorders. It can affect arms or legs, and it has a major quality of life impact for those afflicted. There are very few treatment options for spasticity, which actually spasticity is also relevant to ALS. But the treatment options really are oral medications such as baclofen, injected botulinum toxin, and surgery, where you have to either cut the nerve or the tendon. There are numerous conditions that result in spasticity. On the left, we have a number of neurological disorders that are associated with spasticity. The big one is stroke, and 31% of patients who have had a stroke end up developing spasticity. On the right, we have a number of neuromuscular diseases. ALS is there, and about 80% of people with ALS develop spasticity somewhere throughout the course of the disease process. We've developed MyoRegulator as a first-in-class non-invasive treatment modality. This is the world's first device for non-invasive treatment of motor neuron hyperexcitability disorders. For full disclosure, MyoRegulator is an investigational use medical device only at this point, and it's still currently under development. The picture you see here is our pre-commercial device that is currently in clinical trials. And this is a microprocessor controlled unit that delivers our multi-site DCS technology through disposable single-use hydrogel electrodes, which we have engineered. And this device is driven by a rechargeable battery, and it's portable. FDA has labeled this device as a breakthrough medical device, and it's protected by 46 issued US and foreign patents. So what we are doing is non-invasive motor neuron hyperexcitability suppression. This is simple point of care therapy where electrodes are placed at multiple sites along the neural axis, namely across the spinal cord and on the targeted limb. Low levels of direct current between 1 to 5 milliamps are delivered for between 20 to 30 minutes, depending on the application. And treatment is repeated for several days for initial therapy. The multi-site DCS technology has been shown in animal studies so far that it drives current across the spinal cord and down the peripheral nerve of the targeted limb. So current flows in two directions. And by doing so, it suppresses neuronal overactivity and reestablishes damaged neural circuits. And we've published numerous papers laying out the science behind all this, which are shown below. We've also looked at the molecular mechanisms of how this technology works. And NKCC1 is a sodium potassium chloride cotransporter that's found on spinal motor neurons. And it's involved in maintaining chloride gradient. In the normal situation, there are normal levels of NKCC1 and low levels of intracellular chloride in the motor neurons. When there's disease or injury to the spinal cord, and we've shown this in spinal cord injury as well as ALS models, the NKCC1 protein gets overexpressed, and this leads to high intracellular chloride, which makes the neurons hyperexcitable. Now, what we have found and what we've published is that after multi-site DCS treatment, the levels of the NKCC1 protein are reduced. They're normalized, and as a consequence, the intracellular chloride levels are also normalized, and the neurons become less hyperexcitable. We've now completed 2 clinical trials with positive readouts and have our US pivotal trial underway for the MyoRegulator device. Our US feasibility study was published in 2019. This was in upper limb post-stroke spasticity, and we found a statistically significant improvement in spasticity and motor function. Our EU pivotal trial was completed last year at France's Paris Brain Institute, and this was in lower limb post-stroke spasticity. And we found a statistically significant improvement also in spasticity, motor function, and pain. And we have a very significant paper now being finalized with the results of this study. We now are running our US multi-center pivotal study here in the Boston area at Spalding. This is underway in upper limb post-stroke spasticity, and it's funded by a $4.9 million grant from NIH. And these trials will support regulatory clearance in 2025. So once the US pivotal study is completed, we anticipate FDA submission with targeted launch. And on the regulatory side, as I mentioned, FDA has granted breakthrough device designation to this product, which enables prioritized review and feedback. FDA has also confirmed non-significant risk and that this is on the de novo pathway. Our commercial strategy will be to sell devices plus disposable electrodes, and we plan to partner with leading clinical institutions to facilitate adoption both in stroke and in ALS. Which brings us to ALS. And the science has led us to ALS based on understanding the mechanisms by which our technology works. I certainly do not need to provide a background on ALS to this very specialized audience, but it is a rapidly progressive neurodegenerative disease, and ultimately, this is a disease of motor neuron dysfunction and degeneration. What's relevant to us is that there are 7 FDA-approved drugs. However, none are dramatically altering the clinical outcome or survival. What has emerged in the last 20 years is that motor neuron hyperexcitability appears as an intrinsic part of the ALS disease process. And we have developed a technology to suppress motor neuron hyperexcitability non-invasively. And we believe this provides an entirely new treatment strategy for ALS. So let's talk about motor neuron hyperexcitability. In spinal motor neurons, typically those neurons have a resting membrane potential of about minus 70 millivolts. If it's more negative, it's referred to as hyperpolarized, and if it's less negative, it's referred to as depolarized. Motor neuron hyperexcitability refers to an overactive state where the neurons are more prone to fire because they are relatively depolarized. Both cortical motor neuron and spinal motor neuron hyperexcitability has been documented in the scientific literature as key features of ALS. This has been found in both sporadic and familial forms of ALS, and the whole premise of hyperexcitability suppression in ALS has been supported by recent clinical trials that have looked at potassium channels as well as potassium channel activators and sodium channel blockers. 2 papers have been published, and 1 study is currently underway, phase 1 study in healthy volunteers. Now, we're moving forward based on our mechanism, and we now have an early feasibility study underway in participants with ALS, and we'll get to that. So we've been working over the last several years to develop a robust package of preclinical data that covers electrophysiology, biochemistry, motor function, and survival. We'll get to some of that in a bit, but we've really been working to build momentum in ALS. Some of the data has been presented at leading ALS conferences, including the ones shown here. We've received NIH grant funding for our technology in ALS. Last year, we were very honored to receive the SURF Medical Prize for Medical Electronics in ALS. And this year, we're again very honored to receive funding from FDA to immediately take this product into ALS clinical trials, which we'll get to. To give you a sense of the studies we've been doing, I'll just go through a few slides of preclinical findings. The SOD1 G93 mice developed tremors, paralysis, premature death, and are one of the gold standard preclinical models of ALS. On the left, you see the multi-site DCS setup that we use for animal studies, with 3 spinally electrodes positioned along the spinal column and 2 peripheral electrodes positioned along the sciatic nerve and a returning electrode positioned on the abdomen. On the right, you see the mouse holder that enables the mice to be stimulated while we can elicit stretch reflexes and measure EMGs. With NIH funding, we were able to develop a 4-channel multi-site DCS device for these ALS animal studies. And the interface to it is shown on the right, where we could control the current being administered through each of the 4 channels for whatever duration of time. And this device has been used for the studies that we've been doing in animal models. So the SOD1 G93 mice in our lab develop disease on average at day 113. And after disease onset, there's increased muscle activity that's apparent on the EMG trace, which you see on the left. When you turn on stimulation with analytical multi-site DCS, you see immediate reduction in spike amplitude. And after 3 consecutive sessions of treatment, 60 minutes a day, you see even without stimulation being applied, you see decreased spike frequency and decreased spike amplitude. We also have done sibling studies looking at motor function in these animals with and without treatment. And these are sibling mice that are born in the same litter to the same mother. And they have comparable copy number of SOD1 gene. And I'll start the video and I'll get into a bit more detail. So you see 4 mice here, and the 2 white mice that have not been shaved on their back did not receive any stimulation. However, the 2 mice that have their back shaved did receive stimulation, and they received 5 days of stimulation, 60 minutes per day.
Oh.
So what I think should be apparent here is that there's a significant difference in motor activity between the treated and untreated animals. And we've done a number of motor function studies that are going into a paper that's going to be published in the forthcoming months. So on the molecular level, we've been also looking at specific molecules that have been implicated in the ALS disease process. HSP70 and SOD1 are 2 key proteins, and we've shown that we can modulate levels of these proteins non-invasively using our approach. HSP70 is a chaperone protein involved in degradation pathways, and we found that HSP70 levels in the spinal cord were significantly reduced in carrier non-stimulated mice. However, after we treat with our technology, there is an increase in HSP70, and actually about 150% increase in this chaperone protein. On the right, we have SOD1, and SOD1 aggregates, of course, are a pathological feature in a subset of ALS. About 2% of ALS is SOD1. And we found that while the carrier mice that are unstimulated have high levels of SOD1, which is shown in figure A here, after stimulation, you have lower levels of SOD1 protein in the spinal motor neurons, and we're seeing a 53% decrease. So this is the first time that non-invasive neuromodulation has been shown to be able to impact protein expression in ALS models. And as I mentioned, a paper on these findings has been submitted and will be coming out in the coming months. Beyond looking at changes in molecular markers, we also wanted to look at survival of the animals. And here we have a therapeutic stimulation paradigm. And we have mice that are stimulated and mice that are non-stimulated. And the mean survival time in the unstimulated control group was 11.1 days after disease onset. And in the group that was treated, it was 20.5 days, which is an increase of 84% in mean survival time after disease onset. And this was a statistically significant difference. So this is what we have found. And again, this has gone into a paper that will be coming out. So what we've really been focused on over the last few years is translating into the clinic a new ALS therapy. And as you could see, we've been working on preclinical ALS studies. We've done platform engineering to develop the technology. We've done human clinical studies in other indications related to motor neuron hyperexcitability. And we've been working to get into a first-in-human ALS clinical study. And we're very fortunate to have the clinical expertise of 2 wonderful collaborators. One of them is Dr. Leon Morales-Quezada, who's at the Spaulding Rehabilitation Hospital and is a noted neuromodulation expert. And we're also working with Dr. Sabrina Paganoni, who is also at Spaulding, as well as the Healy and AMG Center for ALS at MGH, and she's a noted expert in ALS. So we're delighted to have their involvement, and with their expertise, we've been able to design the clinical studies. We've prepared FDA pre-submissions. We've received and implemented feedback we've received from the FDA on those pre-submissions. We've received IRB approval to initiate, and we announced the ALS early feasibility study last month. We're grateful for the support that we've had from a number of funding agencies, including MDA, SURF, and NIH. And without their support, we would not be able to move this technology forward into trials. So at this point, we're very excited to have gotten to the point where we have now launched our first clinical trial for ALS. And just to give you an overview, the objective of this first early feasibility study is to assess the safety and feasibility of this device to treat people with ALS. This is a single center, single arm, open label study, 5 subjects initially, and it's a 10-week trial, and there are 16 visits over that period. And this trial is taking place at Spalding here in the Boston area. There are 2 phases to this study. In phase 1, it's the first week. We do 3 treatments done every other day, alternating between cervical stimulation and lumbar stimulation. Then there's a week of rest, followed by phase 2, which is 4 weeks of treatment, where again, we're doing treatment every other day, alternating between cervical and lumbar stimulation. All the treatment sessions last 1 hour, and there's 30 minutes of stimulation on the left side and 30 minutes of stimulation on the right side. And afterwards, we do a follow-up at 4 weeks post to measure the various clinical endpoints. In terms of inclusion criteria, we've kept the inclusion criteria broad to enable rapid enrollment of this important trial. We ask participants to be willing to refrain from participation in other clinical studies during this 10-week period, of course. But again, we've kept the inclusion criteria broad. In terms of exclusion criteria, the exclusion criteria are participants who are on permanent assisted ventilation, have implanted pumps, or have been received injections for botulinum toxin for spasticity, or have implanted devices or hardware in the path of stimulation. What's important to point out is you can stay on standard of care that you're already on, so we do not ask for that to be stopped. In terms of endpoints for this first-in-human study, they're safety and feasibility. So that's what we're primarily looking at for primary endpoints. Our secondary endpoints will look at preliminary evidence of effectiveness. And for this, we're looking at endpoints that this audience is probably quite familiar with, and these are the ALS, FRSR, Rhodes, and ALSAQ-40. For people who are interested in learning more about this study, we could refer you to the Mass General Brigham website. They have a Rally website, and it has information as well as a mechanism to provide your contact information for those interested in actually participating. And this could be accessed through our website if you go to our clinical trial page. So we think there are a number of advantages really to this new approach to ALS. First of all, this is non-invasive, so it does not require surgery or device implantation. It's patient-friendly because it does not require focused attention or movement, really. Home use is going to be important, and our next generation device will enable treatment in a home use setting. As this is not a drug product, we're avoiding numerous issues, and one of them is blood-brain barrier permeability. Some drugs cannot get across the blood-brain barrier, so it's very hard to deliver them to the spinal cord or brain, and we don't have that issue. We also avoid off-target drug effects, and these are side effects of drugs, because we're delivering very targeted stimulation to the sites that require it. We're also avoiding drug-drug interactions, and we're avoiding invasive administration routes, such as injections into the spinal cord. And ultimately, we also view this as synergistic, and it's going to be synergistic with pharmacological modalities that are already being used and are in development. So where are we going with this? And I wanted to lay this out again, given the technology focus of this group. Our next generation device we've already designed. This will allow for multi-level spinal stimulation and bilateral peripheral stimulation. And this will be important to facilitate the efficient treatment of ALS. Our next generation device will also have connectivity to enable home use to provide for better comfort. And we expect to be able to have a wearable device in the future. And we think that will facilitate treatment in ambulatory settings and even at work settings. So this is what we're working on. We think early intervention will be important. And with this kind of technology, the treatments could be started early to potentially preserve motor neuron function. And ultimately, we think we could get to targeted treatment that as soon as the disease begins to manifest at whatever level, we can begin to treat at that level and the surrounding levels to slow the disease's propagation across the neural axis. So that is really what we're working towards. We're fortunate to have a veteran leadership team and a world-class board of directors. Our Chief Technology Officer, Jerry Jennings, is on this call. And Jerry has brought 7 medical devices to approval. Our VP of Regulatory and Clinical Affairs used to run global regulatory at Boston Scientific, and she was at FDA reviewing these kinds of products earlier. Our Chief IP Officer is a 40-year patent attorney who has helped bring this portfolio of IP really to fruition. And we have really a world-class board of directors, and this includes our chairman, who is the former president of Medtronic Diabetes. We have Terry Bresenham, who's our chief innovation officer, who was the chief innovation officer at GE Healthcare. And we have Dr. John Donahue, who was the former founding director of the V Center for Neuroengineering and one of the world's leading experts in neurotechnology. So really to close, what we're doing is working to restore pathways to health. Our MyoRegulator product represents the first neuromodulation device to treat disorders related to motor neuron hyperexcitability. It represents a new treatment modality that's synergistic with existing modalities. It represents an expandable platform that is now focused on ALS. And we think it represents new hope for patients. So we've been able to move this technology forward really with the very gracious help and support of numerous organizations, including the ones listed here. And we cannot do this without the support of the ALS community to help bring this forward. So I know I covered a lot of ground, and I'm happy to take some questions, and I could always be contacted at the email address shown here. So thank you.
Thank you so much. I think we're ready for our first question. What is statistically significant? From Eric.
So in terms of what is statistically significant, I'll refer to the clinical results we have in post-stroke spasticity, and then I could touch on what's statistically significant in our animal studies. So we've completed 2 clinical trials in post-stroke spasticity, and both studies have shown a statistically significant effect on reducing muscle spasticity. So we've shown that we can reduce it in a statistically significant manner. Now we have not. We have just started our first ALS trial, but we have shown it in post-stroke spasticity. In terms of preclinical results, again, there's a paper that's been submitted. We have statistically significant results on numerous readouts, including the biochemical markers, including motor function, including animal survival, and including motor neuron survival. So all that will be published soon, and will be out soon.
Thanks, Dr. Yaghoubi, for your very informative presentation. Our next question is, can this technology slow or reverse muscle atrophy?
That's a great question. And I would say that we do not expect that it's going to reverse muscle atrophy. I think this intervention, what ultimately it can do is to help preserve motor neurons in the spinal cord. And we could lessen probably the emergence of muscle atrophy. But I think once the atrophy is there, I am not going to make any claims that this approach is going to be reversing atrophy. We think we could slow disease progression ultimately, but not once there's atrophy.
We have another question. Paul asked, how is this different from a machine that provides transcranial direct current stimulation?
Yeah, sure. So transcranial direct current stimulation is a similar technology. However, the targets of stimulation are the brain. So they've used that for treating various parts of the brain. What we're doing, actually, there's no stimulation of the brain that we're doing. Our stimulation is applied along the course of the spinal cord and the peripheral nerves associated with the upper and lower limbs. So in terms of the waveform and the type of stimulation, they are similar. However, they're different locations. And I also have to say that what we're doing, multi-site direct current stimulation, is not single point stimulation. We're able to send current flowing in multiple directions to modulate circuits and pathways. And that is not what transcranial direct current stimulation does.
Is there research for this device as an application for non-SOD1 patients?
So at this point, we have a clinical trial, and it's really in all types of ALS, so it is not confined or restricted to SOD1. In terms of the preclinical data that we have generated so far, it's been in SOD1 G93A models. However, we have started on a second model after discussion with numerous people in the ALS field. So those studies have began, and it will be exciting to see the results of our stimulation in the second important model of ALS.
An iPad user, I'm not sure who asked this question, asked if spinal stenosis cause ALS.
That really is, it's not really a question for me. I would refer to the ALS clinicians and experts. So I will defer on trying to answer that question.
And then one of our participants, Lynn, he said that he is an electronics engineer in Houston, and he was wondering if there was any way that he can help.
Well, that's a very gracious offer, and we'd love to, talk and find out more. Our Chief Technology Officer, Jerry Jennings, is also an electrical engineer, and at the heart of this company is electrical engineering. And that is what we're doing. So perhaps I put my email in the chat box, and if you could send us an email, we'd be pleased to have a conversation and see how we could engage you.
Our next question is from Paul, and Paul asked, would taking potassium help with the sodium-potassium differential?
So I don't. That is not something that I would encourage. I think what we're talking about is extracellular sodium as versus intracellular sodium, and I certainly wouldn't advise taking high levels of sodium to try to change that gradient. So that would be my initial thoughts on it. It's a good question. And, I'd have to think further about it, but my initial reaction is that you wouldn't want to try it that way.
Must treatment be given indefinitely, or is a few rounds sufficient for lasting reduction of hyperexcitability?
Yeah, great question. So we're viewing this as ongoing maintenance therapy. We think the more treatments that are given, the better those neurons will be able to preserve their function. So in the trials that we've done for post-stroke spasticity, there have been 5 consecutive days of stimulation. We view that really as initiation therapy, and we think ongoing maintenance therapy is going to be important. We certainly think that will be the situation also for ALS. That's why we're working to get to both a home use device and potentially a wearable device in the future.
We have quite a few questions, so I'll just keep them coming if you don't mind.
Sure.
In the 84% of mice that increased their survival time to 20.5 days, were the mice on continual treatment to the time of death?
No, they were not. And just to, let me see if I could put that slide up. Right. So with those studies, that was not continual treatment. That was a certain amount of treatment. I believe it was 10 days of stimulation.
Have you conducted studies in other mouse models of ALS?
Great question. And as I mentioned, we have just begun the studies in the second model that we've been advised to look at. So mouse colonies being established, and those studies are going to be going on over the coming months. So we're very excited to get that underway.
And Helen says she's sorry she missed the first section and the first couple of slides, but the device looks similar to something that she already has. Is it similar?
I wouldn't know what exactly she's referring to. So Helen, if you could perhaps tell us a bit more, we could comment on it.
I think she said H-wave and TENS.
Oh, I see. I see it in the chat. H-wave I'm not familiar with. TENS, the electrodes look perhaps similar. However, TENS is not able to modulate the activity of the spinal motor neurons. So that's used for very superficial stimulation. So what we're doing really is driving current across the spinal cord and down the peripheral nerves. This has not been done before. We're doing this simultaneously in a way that is able to modulate the activity and firing of the spinal cord motor neurons, and it's their ability to carry a signal.
You. Our next question is about clinical trials. Is there a time frame on how many years someone has since diagnosis for the clinical trial?
So for the clinical trials that we have designed and that we have started, the number of years that someone has ALS is not one of the inclusion or exclusion criteria. So as I mentioned, we've tried to have broad eligibility criteria to facilitate enrollment.
And Jesse asks, how soon do you think the treatment will be approved by the FDA?
Well, that's a great question. And for technologies like this, really, it really depends how fast we could progress through the forthcoming steps. And really, that comes down to availability of capital and support for what we're doing. We have this early feasibility study that we have now started. We have to get that done. We've also designed an expanded feasibility study that brings in more advanced biomarker readouts. Once we have all that data, we're going to have to do a pivotal study, multi-center pivotal study. And then, it'll require regulatory approval. So all this is ultimately gated by capital. And we are very aggressively working to bring in necessary capital to move this forward as fast as it's possible.
Could this product be available out of pocket, and do you have an estimated cost?
So the device right now, it's an investigational use only device, so it is not available. At some point, we expect to have FDA clearance for this device. And once that happens, theoretically, the device is available. However, that first initial approval will be for post-stroke spasticity. So I think it remains to be seen about the cost. I think it's probably premature to get into that at this point. But we are working to move this forward as fast as humanly possible.
And our next question comes from Mark and Paula, and they would like a little more explanation of how hyperspasticity presents in patients, please. Is it only random muscle movement, or is it also something like cramping of hands and fingers?
Yeah, well, as a disclaimer, I am not a clinician working with patients at this point in my life. Spasticity can present in various muscles. However, it could start in the hands, it could start in the legs, and really depending on the disease, it can emerge at various points. So, there isn't a typical pattern or timing to it. It is variable from my readings and my discussions with the ALS clinicians who treat this.
Can this work on upper motor neurons too?
That's a great question. And upper motor neurons are the cortical motor neurons in the brain that synapse on the spinal cord motor neurons. So right now, our treatment is focused on the spinal cord motor neurons. As I mentioned, the electrodes that we place are placed along the spinal column and on peripheral nerves. So we are not working on cortical stimulation. So that is not what this device and the next generation of the technology is focused on at this point.
And Thomas asks, when and where can we expect the publication of the results?
That's a great question. Paper has been submitted, and we have to wait until it goes through all the steps of review and publication. And I would put it in the. It's always hard to say with these things, but I'd put it in the next few months.
Can you please readdress if this treatment is intended to slow the progression? And furthermore, is there a goal for this to induce reversal?
So I will say that we're hoping to go beyond the treatment of spasticity here. And I say that based on the mechanism of action and also the animal studies and the results that we have, where we've shown impact on survival of the animals, and we've also shown impact on motor neuron survival. Our hopes are that it can impact survival and disease progression, but that remains to be seen, and that will only come, on once we run the appropriate clinical trials. So currently we have the animal data, some of which I've shared with you, and we have the results in post-stroke spasticity. So, we're advancing this as best as we can.
Deborah wants to know, is there any hope for West Coast trials?
I hope so. I think for subsequent trials that we do beyond this early feasibility study, we're definitely going to need multiple sites. That for our pivotal trial, it's going to be a multi-center pivotal trial. So yes, I think that certainly is going to be something in the works.
Since this is an external stimulation, would it conflict with a drug trial such as Healy?
So, as I mentioned, standard of care and existing approved drugs that people are taking for ALS are not exclusion criteria for this study. However, we would not want participants to be enrolled in two trials at the same time. So this is a 10-week trial, and during the 10 weeks of the trial, we think it is best for no other trials or no other interventions to be administered.
And Jose asked, so would this feel like an EMS machine?
But by EMS, what exactly are you referring to?
I'm checking the chat to see if he responds. No response. I'll go to the next question, but we'll circle back just in case. Maybe they had to step away for a moment. So the next question, does this machine need FDA approval? And that's from Paul.
Absolutely. This machine clearly will need FDA clearance. And perhaps, Jerry, could I ask you to say a few words regarding getting to FDA approval for the audience? Jerry Jennings is PathMaker's Chief Technology Officer.
Sure. Well, of course, it is a device that is a medical device. It does require FDA clearance, and we have already initiated discussions with FDA with regard to its use in our first ALS feasibility study. It is a pathway, and all medical devices have to go through this pathway for approval. So it can sometimes take much longer than we would like, but that is the nature of a regulated industry.
Thank you, Jerry. Appreciate that.
Hyperexcitability usually happens on upper motor neurons in MND. Does your treatment help to ease upper motor neuron hyperexcitability?
So as I mentioned on a previous question, we're really focused on lower motor neurons here. And, there is axonal hyperexcitability also documented in ALS. And we think that our intervention can impact the hyperexcitability this is lower motor neuron hyperexcitability.
And Andrew asks, have you been able to record peripheral nerve action potentials in response to spinal stimulation in mice or man?
That is a question that's best fielded by our scientific founder, who's not on our call, so I will defer on that.
Going back to the EMS machine, I think Jose said electric muscle stimulation. So would it feel like that?
Yeah, most likely it might be referring to functional electrical stimulation, which is the stimulation of nerves that are attached to various muscles to trigger a contraction of the muscle. And what we're doing, multi-site direct current stimulation, is entirely different and distinct from that. We are not trying to induce the contraction of muscles. What we're doing is we're driving current simultaneously in two directions. One is across the spinal cord, and two is down the peripheral nerve, leading to a targeted limb. And by doing so, we're able to modulate the activity of the spinal motor neurons and impact the circuits and pathways in the spinal cord.
And John Michael asks, where can we find out more?
Well, I would certainly invite the interested members of your organization to take a look at our website. We certainly have lots of information there. Our publications are listed, and if anyone's interested to take a look at those, they're available. There's also more information on our clinical trials through both our website and on the Rally website. So I would say those are probably the main places for information on this.
Could you please explain bulbar efficacy? The participant doesn't really understand since signals pass through the cervical spinal cord.
Yeah, So as I was explaining, we're really targeting the upper and lower limbs at this point. So we are not really focused on trying to intervene in bulbar ALS at this point. Again, we have to start somewhere, and we believe that targeting the spinal motor neuron pools associated with upper and lower limbs are really the way to get started with this approach. So we are not right now working on bulbar forms of ALS.
Does anyone have any more questions they'd like to pop in the chat? I want to make sure we got everyone. No more questions?
Tom, did you have a question? Yes. Tom's writing it in the chat. Is it there?
I see it. So at this point, Tom, for the early feasibility study, we're trying to avoid situations where there is implanted cardiovascular circuitry, really. So I would say for this EFS, we don't view that as being able to be eligible. In the future, it'll require discussions with the cardiologists and the neurologists, but at this point, I would say no. And just to add some color there, we're going to be driving current across the spinal cord, so it's going to be going from the spinal anode to the abdominal return cathode. So we really wouldn't want any devices in the path of current flow that could potentially be, impacted. I think this is out of safety.
And Helen didn't have a question, but she did have a comment, and she says, "Thank you." She's looking forward to the results.
Well, I'd like to say really thank you to everyone for taking the time to hear about what we're doing. So we're honored really to have this opportunity, and we are moving this forward as best as we can. So thank you to everyone for their time tonight on this call. Yes, thank you.